Namaste, this is S.K. Batin Rahman, Faculty of Pharmacy, Jyoti Vidyapeeth Women's University. Today we discussion about DNA replication. In the molecular biology, DNA replication in the biological process of two identical of DNA of one origin of DNA molecule. DNA replication occurred in living organisms, act as a most of the essential for the biological process. This is the essential for cell division during the growth and repairing damage of tissue, while it also ensure the each of new cell of receive its own copy of DNA. The cell is process is distinct property of division, which replication the DNA essential. DNA is made of the double helix, two complex strand. DNA is made of the double helix and two complementary strand. And this is a double helix. Describe the appearance of double strand. DNA has been composed of two linear strands which run opposite each of other twice together to the form. During the replication, this is the standard and separation. Each of the strand of original DNA molecule, each of the several template for the product of the concert. This process is referred to the semi-conservative replication. As a result of semi-conservative replication of new helix of compounds of the DNA strand will be newly synthesized strand. Cellular proof of the error check of the mechanism ensure the nearest perfect DNA replication. In the cell, DNA replication being specific location or origin of the replication of the genome, which contains the genetic material of the origin. Unwinding of DNA origin is synthesis of new strand of unit of enzyme known as helicase. Result, replication fork are the growth of the bidirection from the origin. A number of protein associated with replication for help of the initial continuous DNA synthesis. Most of the prominent DNA polymerase is synthesized new strand of binding of nucleotide. Complement is DNA replication occur during the S stage of metaphor. DNA structure. DNA is an extension of double strand structure. Both of strands are coiled between the form of characterized double helix. Each strand of DNA chain of 4 types of nucleotide. Nucleotide in DNA consists of deoxyribonucleic acid of phosphatonucleotide. This is the 4 type of nucleotide concerned to the 4 nuclear base of adenosine, cytosine, guanine, and thymine. Most common abbreviation ACGT, adenosine, guanine, purine, base while cytosine is thymine, pyrimidine, and the nucleotide has been phosphodiester bond. Create a phosphate diester backbone of the DNA double helix in nucleotide, potentially inward. This is the opposite strand. Nuclear base of the match between the strand through the hydrogen bond and base pair. Adenine is the pair of thymine, 2 hydrogen bond, and the guanine is pair of cytosine is 3 hydrogen bond. DNA strand of directly or direction or end of the strand is called 3-phase or 5-phase of DNA. Okay, this is the DNA exists of the double strand, both of the together of double helix bond. Now, in molecular biology, in biochemistry, we discussing in these classes the DNA replication process is the initially double identified DNA of the origin of DNA molecule. DNA replication occurred living organism act as essential biological initiation. This is the replication process which copy the any kind of DNA molecule in this biochemistry. So DNA are made of the double helix. Complementary strand is helix described of the double strand DNA composed of 2 lines of the strand run of the opposite each other twice together or formed during the DNA molecule. Okay, so DNA in here is DNA polymerase. DNA polymerase are family of enzyme is carry out all of the form of DNA replication. DNA polymerase is generally initial synthesis of new strand can be existence of DNA or RNA strand with the temporal strand with beginning synthesis of fragment of RNA called primate. It must be create a pair of strand template of DNA strand. So DNA polymerase family enzyme is carried out by DNA replication. Replication is but it cannot only system of the DNA strand pairing and temperament strand of beginning of synthesis of fragment of DNA called primate. Okay, this is a DNA polymer of family of one type of enzyme. Just remember it. It's an enzyme because of you say that DNA polymerase. You know that in every word you have a is denoted by the is. It's a enzyme. It's an enzyme. This is a carry out the form of DNA replication. It's a main component of DNA replication, following by initial DNA replication. Okay, the DNA polymerase is added new strand of DNA existing of 3N2 and existing of nucleotide chain of new nucleotide match of the temple strand of the time of create phosphodiester bond. Okay, it's a phosphodiester bond. The energy is phosphate DNA polymerase came in hydrolysis higher energy phosphorylation bond between the 3 phosphate atoms of the each unincorporated base. 3 base are the attached phosphate group called nucleotide. Particular based on 3 attached of phosphate group of called nucleotide triphosphate. When nucleotide are being added to grow of DNA strand is form of the phosphodiesterase bond between the apomix of the phosphate nucleotide of group of accomplice of hydrolysis higher free energy phosphate release of 2 diester bond of phosphorylation enzyme hydrolysis result of phosphorylation into the inorganic phosphorylation conjugate second order of phosphorylation strand within. In the general DNA polymerase are the highly accurate and the initial error of the less mistake over 10 nucleotide. In addition, some of the polymerase also proliferate addition in remove the nucleotide addition of grow of the correct mismatch of basically final post of replication mismatch of replication and mechanism may monitor the DNA error. Being the capable of distinction matching of the newly synthesized DNA sequence together and discriminated the step of replication, final step of replication. The rate of the DNA replication living cell is the first measurement of the rate of the T4 DNA elongation phase of initial E. coli. During the period of existence of DNA increased the 37 degree temperature rate of the 749 nucleotides per second. In mutation rate of the pair of during the T DNA synthesis 1.7 per 10 to the power 8 second. Okay, now we briefly discuss about in here. You see that the DNA existing a double strand and the both of the stranded are together it form double helix. Each of the strand of DNA of chain of 4 type of nucleotide in DNA concerned with deoxy sugar or phosphate sugar or nucleotide. 4 of type of nucleotide concerned with nucleotide adenosine, guanine, cytosine, and thymine, commonly adversing of ACGT and the adenosine, guanine, purine base in this cytosol, thymine, and pyridine. Nucleotide form of phosphate diester bond is created phosphate double helix backbone of the DNA double helix of nucleotide potential inward. Nuclear basic match of the between the strand of hydrogen and base pair, which is the adenosine pair of thymine, 2 hydrogen bond, guanine pair of cytosine, double strand of indirect, and there is different end of signal is called 3' to 3' and 5'. By the conversion of phase sequence of the single strand DNA, it is the left end of sequence of 5N. The right end of the sequence is the 3N. Strand of double helix anti-parallel of the beginning of 5' to 3'. Opposite standard of 3 to 5' is the term of carbon atom of the deoxyribose. It is the next of phosphorylation chain of atoms. Direction of concern in the DNA synthesis because of DNA polymerase synthesis DNA only by direction only for nucleotide 3 into DNA strand in paired with complex DNA. System means that information is contained each of the strand of required phosphodiesterase bond of the strongly hydrogen bond. In actually job of phosphodiesterase bond of DNA polymerase connect to the 5 carbon of nucleotide of 3 carbon of another nucleotide, which is a hydrogen bond of dual helix across the bond of concern. Thank you so much for hearing.